Hi there, and welcome back to Beyond Networks, the evolution of living systems. Last time, we sort of started a discussion about causation by giving a quick overview over what people thought causes are, naturally starting with Aristotle's account of his four, what are really big causes rather than causes, four different ways of answering four different questions about a phenomenon. That was the material cause, what is it made of? The formal cause, what does it look like? What makes it what it is, basically. The efficient cause, what is the process that generated it? What is the agent that generated it? And the final cause, what is its purpose? We then left Aristotle behind and moved via Hume and regularity accounts of causation to the idea that causes could be seen as difference makers in what I call counterfactual theories of efficient causation. I introduced Lewis's theory, counterfactual theory, where if X is not happening, then Y is not happening. So that's a counterfactual conditional statement, and that gives you the gist of what is thought to be a cause. And in more practical terms, we moved on to Woodward's interventionist account that said there's a variety of a counterfactual theory that says if you can intervene on a specific variable in a system and you produce an effect, then that variable is causing the effect. Okay, we enriched Woodward's account with his own terms. This idea of causal stability, a cause is stable if it happens across a broad range of different circumstances. The proportionality that a causal explanation should not include too much detail, but also not too little. It should be at exactly the right level of explanation. And then a bit more complicated, the concept of causal specificity, sort of, that there is a very fine-grained mapping between the cause and the effect, describing the causal influence, and that this sort of specificity means that by fine-tuning the cause, you can also fine-tune the effect. Okay, and I showed you how this applies very straightforwardly to the approach we use in classical genetics. The intervention is we mutate a gene, and then we look at a population of individuals and try to identify the specific and actual difference makers in that population of, for example, white and red-eyed flies. This is easy in systems where a gene, very few genes are acting together and have very specific effects, but it gets very difficult if you have a complex trait where many genes interact in nonlinear ways and where these interactions are important, just like in developmental evolutionary biology, for example. So how, and this is what we're going to discuss in today's lecture, how can we measure the flow of causation through a complicated developmental process? Okay. And so before we do that, we need to build up a bit of toolkit, yet again, I'm sorry. And the toolkit we need here, we need to finally talk about this very difficult concept. And that is the concept of information, because causal specificity has something to do with information. And so we can go back 80 years and look at where it all began. Here in Austria, Erwin Schrödinger, a theoretical physicist who wrote this very famous book, which was simply called What is Life? The physicist's approach to the subject, with an epilogue on determinism and free will. A very interesting book, by the way. And so Schrödinger, I love this picture with the chicken and the egg. It has nothing to do with the chicken and the egg, really. But so it's actually an argument that puts side to side two very different views of how you get order in living systems. And the first view is sort of the order from disorder, the idea that you get what we would nowadays call self-organization. But at the time when Schrödinger was writing the Signs of Complexity, didn't exist yet. And so the only thing he had was statistical mechanics, where you look at a gas and the order arises from the average behavior of large ensembles, large numbers of chaotic microstates, where most of the details about the microstates don't matter. Okay, so you get order because there's a large averaging effect over really large numbers. But he said, Schrödinger was contemplating organisms, and he said, genes are way too small to be in such a large number regime. We don't have that many genes. He made estimates in his book at the time, long before genomics. And he was arguing, because he was a physicist, quantum mechanics through stable molecular bonds ensures rigidly ordered molecular structures, aperiodic crystals. So this was a very powerful idea that actually was very influential for the reconstruction of the structure of DNA, in which small changes, mutations, have big effects. So two conditions here. You have to have a rigid structure. And what he calls an aperiodic crystal is a non-repeating crystal, just like a strand of DNA. And small changes can have big effects on the phenotype. I mean, it's perfectly prefiguring the idea of the DNA double helix here. These aperiodic crystals provide information in form of negative entropy through a code script, which is necessary and sufficient for life. So this is the central idea here, a code script. And this idea has dominated a lot of the research in genetics and molecular biology since then, of course. Not to everyone's benefit, as we will see. So, but this is a very powerful idea, and it's rooted in physics that was, of course, coming forth from Vienna as well, just a little bit earlier. And that, basically the argument that Schrödinger is giving us here is that life is coded in thermodynamics, in chemistry. Information is represented by negative entropy. And so this idea of the connection between entropy and information goes back to Ludwig Boltzmann. Here he is on the Central Cemetery of Vienna on a wintry day, and you can see the equation up here for entropy. And so very famous work. And it was very, 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 very important. I cannot stress this enough for the subsequent sort of discoveries of the DNA double helix and the genetic code. So this idea of a code script being what is causing the form of organisms, this only really applies in the case of the genetic code. So here you can directly and formally see that there is a code table. You can translate one into the other. And so you can apply this sort of ideas about entropy, negative entropy that Schrödinger was talking about directly to the genetic code. But outside, it's much more complicated. So what we're going to look at is how we can look at information in living systems and how that connects to this idea about causation that I was presenting in the last lecture. It's very, very intricately connected, even if it's not clear quite yet. I was telling you in physics, causation is often the transfer of matter or energy. In biology, it's often represented by the transfer of information. So we need to ask the question, what is information? And this is one of the most, complicated topics. I mean, by now, following these lectures, you will have realized that many, many concepts in biology have multiple meanings. They're context dependent. Often people don't quite know what they're talking about. Information is probably the worst of all. Okay, so here is philosopher Peter Godfrey Smith. In 2007, he writes, the use of information is strikingly prominent in contemporary biology. Undoubtedly true. Everybody's using it. It's important. It's a good way to look at causal flow in complex systems, actually, we'll see. But, and this is a point I want to make early and I want to make over and over again, the unnecessary reification of information is something that goes on Godfrey Smith's nerves. Okay, he says, information is not a thing. So here's the second mantra of this course. After structure does not determine function, information is not a thing. It's not even Is it a process? Information transfer is a process. What is information? Okay. So there are people walking around that say, oh, the universe consists of energy, matter, and information. Now, that is what we call scientific bullshit, and we will come back to that topic in a later part of the series. So we have to be careful here. Information is used in two main ways when it's not used often in biology, actually, and this is something I have to rant about very quickly. It's used in ways where the people writing about information have no idea what they're talking about. Okay, so this is particularly bad in developmental biology. We'll come back to the idea of positional information. Most people that mention this term have no idea what they're talking about, and they use it in a very vague, metaphorical, or sometimes just simply incorrect way. But if you know what you're talking about, then there's two main ways of what information can mean. And one is a formal sort of sense of information as an information theory. It's called Shannon information because of the founder of information theory. It's about correlation, communication, and entropy. We'll explain it in a sec. The other idea is semantic or intentional information, and it's very, very different. It's about meaning. It's about reading meaning out of a signal. Okay, the first clearly defined concept is not about meaning. You can measure Shannon information, but it doesn't tell you how much meaning there is in a signal. You'll see. While semantic or intentional information, you can imagine that there's a cell, gets an inductive signal, and it interprets it according to its circumstances. The cell is an agent. We'll see that in a later lecture. So it has an active sort of component in there. Interpretation is an active process. That is all semantic information that the cell is receiving there. And that is not very well defined and not measurable at all. We'll talk about it a little bit in next lecture. So let's focus on the first sort of purely physical interpretation of the term. And here is Claude Shannon, very dapper looking, and the main diagram in his 1948 paper that founded the field of information theory. Information theory is basically concerned with how you can get information that comes from a source through a transmitter, down a line, to a receiver and a destination. And there's a noise source on the line, and so not all the information will arrive here. And the very simple technological quest was to determine how much information gets across, which was measured by correlation between the signal in the transmitter and the signal in the receiver, of course. So one very important feature of Shannon's information that you need to consider is that Shannon information can only be lost through noise. It can never be gained in information theory. And this is a problem in biology. When people started suggesting, right after Shannon invented his theory, to the chagrin of Shannon himself, there's a nice quote where he says, you shouldn't apply this outside the domain that it was intended for. But people apply it and say, okay, we can look at sort of development as a channel from a sender, which is the genome or the embryo, the zygote, and a receiver, which is the adult organism. But if you do that, and here's an early criticism of this view by Michael Apter and Lewis Wolpert in 1965, they set out to do something really crazy. They said, look, there's a very simple condition in information theory. Information can never increase. So let's look at how much information you need to build a body out of predefined parts. And what they do is crazy, because they go and estimate that it will take about 10 to the 25th to 10 to the 28th bits. I'm not going to go into the details. They admit themselves that it's a large range, but it's a large number. Okay, that's the main point. And now they go on and say, okay, so how much information is in a germ cell? And they have another very convoluted argument, and they come up with 10 to the 11th bits. And again, they admit that this may not be sort of a very rigorous scientific endeavor here, but the fact is that the differences between those numbers are so huge. Okay, 14 to 17 orders of magnitude, massive, galactic, huge, humongous, okay, that you can say with some confidence that information must increase during development. So you cannot use Shannon theory, simply apply it to development and say, okay, development is a channel, where the genome is transmitting information to the adult phenotype. So here's a quote: "It is perhaps more useful then, when considering how increased organization and complexity arise during development, to think in terms of the egg containing instructions, of a program of development, rather than as containing information." Okay, so this idea of a developmental program, of course, came out, it was really hot topic at the time because it came out independently in 1961 in a publication by Ernst Mayr and a very influential paper by François Jacot and Jacques Monod, for completely different reasons. They inferred there must be some sort of, like a computer program controlling development. And now, so this idea was supposed to solve this problem. But does it really? Okay, so there's some sort of set of instructions. What are these instructions running on? What are these instructions? What's the software? What's the hardware? There is no distinction. It's even more complicated. The program sort of writes itself, right? I mean, the program runs on the hardware, and it sort of, the program makes the hardware. It's not like a computer at all, right? It's a metaphor that breaks down very quickly if you think about it. But for Wolpert, this was the solution. And so he set out, Louis Wolpert, to generate a very sort of a famous thought experiment, one of the few very famous thought experiments in developmental biology. So let's have a look at that, because it applies straightforwardly to Drosophila. And we're going to look at Drosophila in the next two lectures very carefully to sort of apply the ideas we're developing here in the real world. So remember that early patterning, segment determination in Drosophila happens through the cascade of the segmentation gene network. And in particular, there's a first step here where you go from these sort of very broad maternal gradients to these sort of overlapping sets of gap domains. I'm not going to go over this, just rewatch the earlier lecture that covers this if you're not familiar with the system. And so what's happening here is that these maternal gradients, they act as classic morphogens. So a morphogen is, in a general sense, is a sort of a substance that generates form. But in this particular sense, it is a long-range gradient that emanates from some source at one end of a tissue. Here's the head of the fly embryo, and then diffuses across this tissue where it's degraded at the other end. And if you assume, now just for simplicity, that there is no degradation of the protein in between, you get a linear concentration gradient of this morphogen. It's not quite right. It's actually an exponential gradient here, but you get the point. So the second step in this model is then that there are specific concentration thresholds in this morphogen that the cells can detect. They can read out how much concentration there is. And if they fall above or below these thresholds, they will switch into different differentiation programs. Remember, these are sort of like computer programs in Wolpert's view, and switch on sort of the blue pathway here, the white pathway in the middle, and the red pathway here. So they will end up differentiating in different ways and implementing this beautiful French flag pattern. So this is called the French flag problem. It's a thought experiment, a toy model that is supposed not to represent an accurate, here's a beautiful illustration of a model used as a tool. It's not an accurate representation of the actual system at all, but it's very powerful because it gets to the essence. So, and the essence here was that Louis Wolpert was claiming that this is how you can get from a program in the genome to spatial pattern. Okay, so he says all of the interactions that you need for this problem to work is that they are encoded in the genome. Okay, so the cells are running a specific program. It tells them how to read these thresholds. So you get from this sort of, he solves the non-trivial problem of getting from a sort of a cellular program that Jacot and Monod were talking about to a spatial pattern. This is the main aim of the French flag problem. And so beautiful, beautiful illustration, model as a tool. It says, this is how you should be thinking about it. And it influenced generations of developmental biologists that study morphogen-based gradient-based patterning, including myself. So what is this about? Wolpert called this sort of the content of this gradient, what the cells are reading. He called this positional information. So he said basically the cells can reduce their uncertainty, the negative, it's a negative entropy here. They can reduce their uncertainty about where they are in the tissue by reading the concentration gradient. Okay, so there's a transfer of information directly from this gradient onto the cells, the target cells in the tissue. And you can actually, so subsequent work, recent work by Thomas Greger and colleagues has shown that you can formalize this. So what they did is they measured the gap genes, the expression patterns of the gap genes. Here are pictures of embryos, head to the left, tail to the right, and it's a sagittal section through the middle of the embryo. So you can see the expression patterns of these different gap genes, staggered different expression patterns. And they measured not only the concentration, but also the variability of the patterns, and in particular, the covariance of the different patterns. So how do they influence each other's expression? They also did the same thing for the maternal gradient. And here you can see these beautifully quantified graphs. They do this all in the same embryo. It's beautiful. It's a very technically difficult experiment to do. Beautiful quantitative work in embryos. And so what they do is out of this distribution from many embryos of expression patterns, you can formalize this notion of positional information that the cell reads out. So imagine that you're at a specific level of concentration here on the y-axis. That's just the distance from anterior to posterior in percent. And so imagine that the cell is reading this concentration here, okay? And so then if you just draw a line through here, you can tell that the cell from reading this concentration could be anywhere along this profile with a probability that's indicated on the y-axis here. So it's a pretty broad profile. It doesn't really know. It's somewhere in this area. There's 2 peaks, but it doesn't quite know, right? And so on and so forth. If you're lucky, you're at a better position. But here you see there's 3 peaks, and they're very widely spaced. So the cell reads this sort of concentration. It could be here, it could be here, it could be here. It doesn't really know. And the same for this level here. So these are sort of the possibilities. And so from these sort of distributions, you can measure, doing some standard information theory, what the information content is of each of the profiles. So, and here are 4 of the gap genes expressed that you saw before in the slide before. And they calculated that each of them contains close to or a little bit less than 2 bits of information. So this is interesting. So imagine the gap gene is just an on-off switch, then it would only contain 1 bit. So they contain almost twice as much information, which tells you immediately that the sort of graded expression pattern is important. But it also tells you that you know, I mean, how many bits of information do you need to actually know where exactly you are? You need more than that. You need more than 2 bits. So they calculated how many bits you would need to know precisely down to 1 nucleus position where you are. And there's about 100 nuclei along the embryo. So you can estimate that you need about 4 bits. And when you add up, when you calculate the average sort of the joint information content of all those 4 patterns, they add up to just about 4 bits. So basically, they check the readout as well. So the parallel genes, or the transcriptional targets of these genes, they contain a little bit more information. Here's the problem again of information going up all the time, but it's on the same order of magnitude. So basically, here you have a system. The information flow through the system is basically almost optimal. Okay, so that's what they conclude, and they have a lot of research into this very controversial topic of whether this network actually does optimal decoding or not. I mean, it's better than optimal. It actually increases information content. And remember, this is not something a decent information transmission system should do. But that's not the point here. The point is that we have a rigorous measurement. We can show that Wolpert's positional information conforms precisely to Shannon's idea of information. So, and it can be measured and quantified in a rigorous way, following the theories both exposed by Shannon and by Wolpert. That's really cool. So this type of information, however, is about precision of patterning. Okay, it tells you how sure the cell can be to be somewhere. Okay, it's also about the propagation of errors through these expression patterns. So it is not how the pattern arises in the first place. And therefore, and I may be flogging a dead horse, nothing seems to be a mechanistic explanation, but this for sure is not a mechanistic explanation. It doesn't tell you how the pattern is made. It only tells you that it's very good. It's very sort of precise, but not how it's actually arising. It also doesn't tell you how the information, the precision can actually increase over time, which is not what a standard information transmission system should do. Okay, so here we are. It's a cool approach. It allows you to check how precise development is, and it gives you new riddles. How does precision increase over time? How does information increase? So information during development not only increases because the adult is more complicated than the embryo or the egg, it also increases because patterning becomes more precise over time, which is amazing. So normal standard systems that physicists study, far from equilibrium thermodynamics, they don't do this. And it's not clear, actually, with all the physics that we have, how this is actually happening mechanistically today. So what we need to explain is, again, how do all these factors work together? And I'm showing you a very sort of stylized representation of the gap gene system that actually indicates is a spatial representation. I'm going to go into the detail later on, but all you need to see is that these boxes represent where the gap genes are expressed, and these interactions represent the interactions between them. The background color represents the maternal gradients, and so you have a very complicated system of activation by maternal gradients and neutral repression by the gap genes, and we looked at this particular interaction here before. So we have a very sort of complicated interaction network, and we need to bring these two things together. Okay, so we can make a dynamical model of this, which gives you this topological explanation in terms of attractors and their bifurcations. And then you have sort of the causal explanations that you get from looking at the precision of the system or from doing genetics on individual interactions here. And this is where information theory can also be very useful. Okay. But before we get to that, before we look in a bit more detail at the network and how we measure information flow in the network, let's revisit this sort of claim by Schrödinger that life is nothing but a code script. Actually, it is not. Okay, so here is Stuart Kaufman, and he has a beautiful sort of piece in a book on Schrödinger, What is Life, the Next 50 Years, where he says, information in code scripts stored in periodic crystals are neither sufficient nor necessary to explain life. Why? Template-based replication succumbs to an error catastrophe. What does that mean? So if you have something copying itself without the self-regulating properties of the organism around it, it won't work. It will make more and more errors. We'll revisit this when I talk about the reproducer perspective on evolution. Okay, so there is no copying of genetic material without an organism, and that organism has very mysterious properties of self-organization, of being able to kill errors in ways we don't understand yet. But maybe it has something to do with attractor states that can be stably inherited even in light of massive genetic and environmental perturbations. Okay, so these sort of systems, remember Waddington's idea of canalized developmental trajectories, these are robust against perturbations. And so it's not just the information in the sense of Shannon that flows from the genes that is producing this precision, this canalization. It can't be. There's more to it than that. Okay, so the vision that Erwin Schrödinger set out, and that a lot of people, the people who are talking about genetic programs even today, are claiming, without having any sort of scientific basis for it, they simply fall short of explaining all the phenomena that we see during development. The tools we have from information theory do not work to explain these phenomena, and they're the wrong tools, because information theory at this level doesn't give you any sort of mechanism. So you get order from this order in biology, and so here's Stuart Kauffman writing, most of the beautiful order seen in ontogeny is spontaneous, a natural expression of the stunning self-organization that abounds in very complex regulatory networks. We appear to have been profoundly wrong. Order, vast and generative, arises naturally, and we have still not the faintest clue how. Okay, so we're at the beginning of this journey. Schrödinger himself was a little skeptical. Towards the end of the book, he writes, from all we have learned about the structure of living matter, we must be prepared to find it working in a manner that cannot be reduced to the ordinary laws of physics. Wow. And that not on the ground that there is any new force or whatnot, not some sort of mystical vitalism directing the behavior of the single atoms within a living organism, but because the construction is different, the organization of the living system, from anything we have yet tested in the physical laboratory. Visionary, okay? Schrödinger goes back and forth between his different views, and he settles on the code script. But if you read the book, and I recommend it, you'll see that he's sitting on the fence. So let's get back to this problem of the network and the flow of information through the network. So what we can do, instead of looking at overall error propagation in expression patterns produced by a network, we can look at how information flows through the network. And this connects back to the topic of causation, because by measuring how information specifically flows through the network, we can infer causal flow. So the principle is quite simple. So what Shannon's theory under the hood is actually based on is a principle of covariation and correlation. Just very briefly, the covariance measures how two random variables, usually linearly, but doesn't have to be, depend on each other. And so if the covariance is 0, there is no correlation. If the covariance is positive, there's a positive correlation, negative correlation. And we have different correlation coefficients, which are normalized measures of covariance. For example, the most famous and most simple correlation coefficient is the Pearson correlation coefficient, and it measures linear correlation. So this is a perfect correlation here. And so as you make the pattern more and more fuzzy, correlation goes down. Here's a perfect negative correlation. But this, is a funny case. It's 0, right? Or these very irregular sort of nonlinear correlations are also not picked up by this correlation coefficient. So nonlinearity is a problem here, but you can use more sophisticated measures. And so this is how we're coming back to this idea. What is information actually? So it has something to do with the entropy of the system. And so the more, the entropy of a random variable measures how unpredictable it is, right? And the more unpredictable a random variable is, the more information it can contain. The more you can have this negative entropy, okay? If there's no entropy to start with, no negative entropy, no information. So imagine 2 coins. If you have 2 times head on a coin and you throw it, there's no information, it's always head. So there's 0 information content. While if you have sort of a biased coin that falls 70% of the time on the head and 25% on tails, it has sort of this intermediate information content. If you have a fair coin or a fair dice, then you get full information. So 2 sides of the coin, you get 1 bit of information out of throwing it. If you throw the dice, you get 2.585 bits, because the die has 6 sides, and you're less and less certain. So it's harder to predict which face will be up with a die than which face will be up with a coin. Okay, so the more uniform the outcome and the larger the number of outcomes, the greater information encoded in the system. Okay, so we're getting there. So information is not a thing. Information is about the potential of different behaviors. And in particular, it's about the reduction of the uncertainty about these different behaviors. So if you have more choices, you need more information to know where you're going, right? But sort of if you take this to the extreme, the sort of process that contains most information, these are completely random processes, like a die. And so the more random the message, the more information, in Shannon's term, there is in the system. This is not a measure of meaning at all. A random process, completely random, is the one that contains most information. So this is problematic. Again, don't interpret this type of information in any sort of semantic way that it carries meaning. It's just about the number of bits that are going through some sort of channel. Okay, so we can make this idea of information and entropy a bit more practical by looking at the mutual information of two different variables in a system. Okay, so the mutual information is a measure of the mutual dependence between two random variables. We don't have to go into technicalities here. It tells us how well we can predict one variable by measuring only the other. Okay, so here again from Wikipedia, a bunch of measures of mutual information, and you can see it captures the sort of non-randomness of the interactions, the dependence of the two variables, independent of the exact nature of the correlation. It doesn't have to be a linear correlation. So these are now quite high, while mutual information is lowest in this case, of course, even lower than here, because you cannot predict from measuring X, from measuring Y here, you cannot predict anything about the value of X. Okay, so let's apply this to a network. So what we take, let's take a sign-directed graph of a network. So let's say this represents some sort of network of causal influences between variables, right? Positive, negative, doesn't matter really. And we want to measure the causal flow. Which one of these interactions are relevant? Which ones are not? How are we going to do this? Okay, and the trick is we can determine the specificity of each causal interaction. We can measure causal specificity. So remember, the task from this big picture in the beginning that I showed you was to pick out those causes that are most specific and are actual difference makers. Okay, I have to remind you of this. So this is the task we have here. And so we can do this by measuring how much mutual information a specific causal variable carries about its effect variable. So for example, let's take this interaction here. We can measure variable number 4 and try to predict variable number 6. And we can determine using mutual information how much information variable number 4 will carry about variable number 6. It will be, it's a tricky case because it's the only activating input into this variable. There's 3 repressive inputs though that will make the situation complicated. Okay, so the correlation here will not, the mutual information will be far from perfect. Okay, but also we need to measure how much entropy is in the biologically relevant variation of the causal variable in the first place. Remember, the die has more information potential than the coin. Okay, so here we need to pick out those factors that actually vary. So these, this is from a paper by philosopher Paul Griffiths and his group, and they called these sort of difference makers specific actual difference makers. They call them SADs, which is, I don't know if that's a good acronym, but here we go. So every node in a network becomes SAD. Okay, it becomes a specific actual difference maker in the context of the network. And we can assign a causal specificity to each one of those interactions. Okay, and we can then pick out which ones are more informative, right? To tell us about certain changes in a network than others. But still, we're not getting the whole picture here, right? So now we've sort of taken information theory and we've used it as a way to measure causal specificity in a way that's very clearly defined theoretically. Okay, so these concepts very clearly correlate with each other. So we have a tool to measure causal specificity, the flow of causality in a network. And we have these dynamical systems models that give us the overall dynamical behavior, the sort of attractors of the system and their bifurcations. So we need to bring them together. And so how you actually infer these causal networks from data is a very important, very big science. There's a beautiful little book called The Book of Why by Judea Pearl about it. And it's exactly about how you infer what are called causal networks. Causal modeling are basically network graphs that have numbers attached to the causal specificity of their interactions. Okay, Judea Pearl, incidentally, is the father of Daniel Pearl, who features in the hyper-normality documentary that I showed a link to in the first lecture. So very mysterious connections here. I don't know. It's a very sad story if you look into it. So Judea Pearl is a massive figure in the field of artificial intelligence and causal inference. And these methods are extremely important for coming up with predictions about how systems actually work. But they are not yet mechanistic explanations. So they are of massive predictive power, but they're, funnily enough, causal graph models are statistical models, even if they're very fine-grained. They're much more fine-grained than the statistical analysis of information theory on the whole level of the output of the system that we looked at before. But they're still not causal mechanistic explanations, right? So we have two problems that remain. The first one is this notion of causation. It is a bit weird. I'm still sort of trying to get my head around it. It is a population level property, okay? You need to measure variation over different individuals. So don't we want to know what's causing something in an individual? And how would we do that? I don't know. We'll come back to that later. And the second problem, of course, we may have heard that before, is we still haven't put the system back together again. We have fine-grained our causal approach so much that it amounts to decomposition, causal decomposition of the system just by other means than when we manipulate and intervene on the system with genetic methods. So in the end, this does nothing else but, it helps us assign causal specificity in more complicated cases than classical genetics, pure interventionist approaches. But in the end, we need to put the system back together again, recompose it using a dynamical systems model. So to wrap this up, we still have this task of bringing together causal decomposition of the system by genetics or causal modeling and models of its dynamics. Okay, and I think that's the crucial task for understanding how the system does what it does. And this is the aim of dynamic mechanistic explanation. And beyond that, as we'll see in the next module. So this was all very abstract. So I hope the connection between causal flow in a network and information transfer is clear. I also hope that you understand now that both the ideas of causal interactions and information flow are very abstract. Information is not a thing. It's not an ingredient of the universe as a constituent, a substance, nothing like that. It is a way of looking at the causal flow in systems. It's a very powerful way, very important, but it needs to be complemented with methods for recomposition. So we will look in the next two lectures on a detailed model of the entire gap gene system, a complex system, and we will examine, using that example, how you bring together the dynamical systems model and a sort of an idea of causal specificity and the flow of causality in a complex system, and how we can represent that and what we learn from that. For now, I hope you will tune in again when we talk about gap genes, a system that I've spent my last 20 years of my life on studying. And I'll be very excited to show you with the example of my own work how we apply all of these ideas that we have learned about so far and sort of bring them finally together. Okay, so I hope you tune in again for that, and I'll see you next time.